So in the lab, when we have our fractionated, what we call our cell lysate, so after we work up our cells, we centrifuge them, we separate the soluble from the insoluble fractions, and then we have our mixture of proteins. We call that the cell lysate. So after we've fractionated the cell lysate, what we actually have are a set of tubes that usually look all the same. They're clear. Like you can't tell them apart by eye. So we need a variety of methods in order to actually detect the protein that we're interested in. And so there's several different ways to do that. So the first general method that works for detecting a lot of proteins is by UV-vis absorption spectrum. So recall from our previous lecture that aromatic amino acids such as phenylalanine, tryptophan, and tyrosine all absorb in the UV around 260 nanometers to 280 nanometers. And so the more of those aromatic amino acids that are in the protein, the larger the absorption will be in that area. And so what you usually end up with for a lot of proteins is this is a UV-vis absorption spectrum where the x-axis is wavelength and the y-axis is absorbance. What generally happens is there's a large peak, for instance, centered around 280 nanometers. But the problem with that is that a lot of proteins, almost all proteins, have some amount of tryptophan, tyrosines, and phenylalanines. And without actually having the purified protein, you can't tell what the difference is in the intensities between these proteins. You can't compare them when it's a mixture of proteins. So this isn't very useful for identifying a specific protein that we're interested in. It's only a general method for identifying proteins. So if we're looking for something more specific, depending on what type of protein we're using, if it binds an organic molecule that absorbs in the UV-vis absorption, in the visible region, so for instance, Heme, such as, which is an important iron-binding organic cofactor that binds to proteins. It gives colors, it typically gives the protein a red color. So a very famous heme protein is of course in blood, which is called hemoglobin. And that is what causes your blood to be red. And so this absorbs out in the visible region at about 420 nanometers. So a picture of the spectrum is also shown here in this UV-vis absorption spectrum, where the solid trace is the spectrum of hemoglobin, or excuse me, myoglobin, which has a heme inside of it. And so we can use this absorbance, if our protein has a heme in it, we can use this absorbance to determine where our fraction might be. And of course, if this is a red protein, we can also kind of see this in the fraction. We would expect to see a red fraction amongst all the colorless fractions. However, there are also a lot of heme proteins. So it still may not be as specific as we want it to be, but at least allows us to, if we can use this, it allows us to separate, for instance, heme proteins from non-heme proteins that don't have this absorption. Now the most specific way is if we know something about the function, which means we have to know. This is not always the case because remember, typically, remember what we're trying to do when we're isolating proteins is try to figure out the function often. But let's say we know something about the function, maybe from some other method. And so, for instance, there's a protein called cytochrome P460. That oxidizes hydroxylamine, this molecule here. The balanced equation is shown on the right-hand side. So in the balanced equation, you see that two hydroxylamine molecules are oxidized and electrons are transferred to an oxidant called dc-pip, which is short for dichlorophenol indophenol. So this is a chemical oxidant. and it absorbs at 605 nanometers. What that means is that this oxidant has a very deep blue color, and we'll very quickly tell you why that's important in a second. So hydroxamine is oxidized to N2O by the cytochrome P460, which is nitrous oxide, and then in the process electrons are transferred to DC-PIP to form DC-PIP-H2. So this is a reduced DC-PIP. And then other products are water and protons. Now, ideally, if we know something about the function, then we can look for the protein amongst these fractions by looking for activity of the protein, for looking for this hydroxamine oxidation activity. And in order to assay something, we need some way to probe that activity is occurring. So DC-PIP is really useful in this respect because it's blue. And then when it's reduced, it's colorless. So that means as electrons are being transferred to DC-PIP, the color goes away. And that indicates that hydroxamine is somehow being converted to nitrous oxide. So in this figure on the bottom left-hand corner is a figure following the consumption of DC-PIP. The asterisk is when hydroxamine was added to the enzyme. And then you can see immediately after hydroxamine was added, we see this decrease in the concentration of the DC-PIP all the way to 0 over about 2 to 3 minutes. And so that was telling us, that tells us that we probably have this enzyme activity. So another way we can do it is instead of looking at reactant consumption, we can also look at product formation. And so nitrous oxide can also be detected very specifically by a specific electrode. And so that's shown in this red trace here, where again, as we add hydroxamine, as indicated by the asterisk, the amount of nitrous oxide increases over time on the same time scale as the DC-PIP consumption. And so in doing this, we have an alternative means to test our fractions to figure out where the protein is. So we can add hydroxamine DC-PIP to these. Oops. Make that a little bigger. We can add hydroxamine and DC-PIP to these and see which ones either consume DC-PIP or produce nitrous oxide. So maybe this one doesn't, this one doesn't, this one, but this one does. And then this one does not. And so that tells us that this is the fraction that we want to keep out of all the proteins that we separated from each other.